Recording? Are we recording? Yes, we are.
Okay, all right. Well, we left off on Tuesday looking at the beginnings of discussing a couple representative signaling pathways. The first pathway, the first two, we're going to use these so-called G-protein linked receptors. These are receptors that directly bind to the trimeric G proteins and activate them. In other words, stimulate their GDP for GTP exchange and turn these guys on. We went through one of them on Tuesday. That was the cyclic nucleotide signaling, where the G protein binds the receptor, it gets activated, and it activates the enzymes adenylate or guanylate cyclase, making cyclic AMP or cyclic GMP, these modified nucleotides with the phosphate twist around the circle. And those, in turn, bound to and activated the protein kinase A, or PKAs, or the PKG family. And those kinases can do all kinds of stuff. Okay, that was one of them. That's where we left off. Now what we're going to do is we're going to look at a second G-protein linked receptor pathway called the phosphoinositide signaling pathway. I know, it sounds like a jawbreaker of a name, but we'll explain what it is in just a moment. Okay, key point about this. This pathway turns on an enzyme that splits a membrane phospholipid into two components. One component is polar, goes in the cytoplasm and regulates things in the cytoplasm. The second one is hydrophobic, stays in the membrane and regulates things in the membrane. So this pathway is widely used because you can cover all bases, cytoplasmic and membrane events, all at the same time. So we see this pathway in a number of different things. For instance, most of our sense organs use this pathway. Vision uses this pathway. Our chemical senses of smell and taste use this pathway. I'm not sure, but I believe hearing does as well. So if you didn't have this pathway, you'd be blind, you couldn't taste or smell anything, you probably couldn't hear, but it wouldn't make any difference because this pathway is also used in sperm egg response when fertilization occurs. In other words, when the right sperm meets the right egg, those pathways triggered off in both. Triggers off events in the sperm cell that are necessary for fertilization, and it triggers off events in the egg cell that are necessary for fertilization. So without this pathway, fertilization wouldn't occur at all. And it's also used in many other physiological and cellular processes. So let's take a look at this pathway. We're going to set things up. Once again, a cell membrane here. We're going to have a receptor. The receptor binds some kind of ligand, or here's the receptor. Okay, now, here again, most of these receptors bind specific molecules, specific ligands, and there's a whole bunch of them around, and a whole bunch of different G proteins that bind to particular occupied receptors and not to others. Some of the receptors, though, also respond to other things. For instance, the photoreceptors in our eye, the rods and cones, they have these proteins called rhodopsins that contain a vitamin A derivative pigment that absorbs light. So in this case, those receptors respond by absorbing light. And we have other things that respond by changes in temperature. Case in point, how come hot chili peppers taste hot? They've got particular molecules called capsaicin in it. And that's the stuff they use in pepper spray to spray boyfriends with or ex-boyfriends and things like that. Oh, yeah, that's really what it's supposed to be used for. Okay, but that's what they have in pepper spray. Okay, now, it turns out it feels and tastes hot because it turns out it stimulates the temperature receptors. So the receptor doesn't realize that it's a chemical, not that it's being scorched, and it responds the same way. As a matter of fact, the two put together give you a nasty response. For instance, one time I'm making a dish and I'm chopping up those nasty, fiery banana chilies. You know, one's about this big. They are some of them are mild and some are just like brutally hot. So I'm cutting these things up, and I feel, you know, some of the oil gets in my skin and burning a little bit and stuff. When I went to take a shower that night, nice lukewarm shower, it felt like I stuck my hands in boiling water. Water wasn't all that warm, but I was already getting my heat receptors stimulated from the chili extract. And when I added some real heat to it, it just made it really downright painful. Okay, so that's an example. In this case, a receptor that a plant substance stimulates the same receptor. And, of course, the plants do this to try to prevent predation, except by people like Indians and Thais. Yeah. You ever had native hot Thai food? Okay, so here we have the receptor binding the ligand. Now a trimeric G protein comes along. Here's the gamma, the beta subunit, the alpha subunit, and that stimulates, once again, these receptors act as guanine nucleotide exchange factors. They're going to stimulate the exchange of GDP. GDP goes out, GTP comes in, stimulates that exchange. Once that process happens, the G protein alters its tertiary structure, it falls off the receptor, and the alpha subunit falls off itself, by itself. So here we have the alpha subunit and GTP unit, and then of course we've got our beta and gamma subunits separate. Okay, the alpha subunit is active. It is now capable of binding to and activating target proteins. And the target protein in this case is going to be something, another large membrane-bound enzyme, and it's called phospholipase, especially if it's spelled right. Phospholipase C, or PLC for short. Now with a name like phospholipase, it tells you that that enzyme does something to phospholipids, which are the major components of animal cell membranes. And there's lots of different phospholipase families around. Most of them attack any phospholipid in sight. Case in point, if you ever had a close encounter with a copperhead, cottonmouth, rattlesnake, or what have you, if it bites you, one of the many things that in the venom that pumps into you are a bunch of phospholipases. They chew up your cell membranes around the site of the bite, causing tissue death, pain, and stuff like that. And the stuff gets into your bloodstream, it chews up the red blood cell membranes, and all the hemoglobin leaks out, and then you basically die. Okay. Now, phospholipase C, though, is unusual. It does not attack every phospholipid in sight. It's an extremely specific family of phospholipases. It attacks only one particular phospholipid. And that phospholipid is just a few percent of the membrane lipid. So if it attacks this phospholipid, it's not going to damage the membrane integrity. So the membrane stays intact in this case because it's got a very limited target. And that phospholipid is known as, here's a jawbreaker, but I'll explain it in a minute. Okay, its full name, phosphatidylinositol 4,5-biphosphate. Usually, frequently abbreviated as PIP2. It's a lot shorter and a lot easier to deal with. Now, what does that mean in ordinary English? Okay, it's a typical phospholipid. What you've got here is you've got a 3-carbon glycerol backbone. I'm not going to draw the whole glycerol molecule or anything. Attached to 2 of those carbons are going to be fatty acid chains, which would represent as this. Now, that's typical phospholipid. Now, phospholipids, you have a polar head group, which includes a phosphate ion. So let's put a phosphate ion here. And then that oxygen of the phosphate is connected to a 6-carbon ring-shaped alcohol called inositol. And that's very polar because, of course, it's got OH groups. I'm just going to represent it as this hexagon here. And there's an OH group coming off of every single one of those carbons, so it makes it extremely polar. Now, not only that, you have 2 phosphates attached over here at the so-called 4 and 5 position. 2, 3, 4, 5. So, of course, those have electrical charges on them. So we have a phospholipid, typical phospholipid, very hydrophobic here, the glycerol and the fatty acids. And this is about as polar as you can get. 3 phosphates and a very polar molecule to boot. Okay, so that's our PIP2. Now what happens, what does phospholipase C do to this? Okay. Think of fanatical Al-Qaeda, Taliban types. Think of Mexican drug lords. What do they do to ones they don't like? They chop the heads off. And that is exactly what phospholipase C does. It cuts the polar head group off of the rest of the molecule. So phospholipase C cuts right over here, does a hydrolysis reaction. Okay. Off with the head. And they put it up on YouTube. Warn everybody. Okay. So now we do this cut here. Okay, and that generates 2 separate molecules. We're going to have this molecule here, the glycerol, and 2 fatty acids. And this, of course, is very hydrophobic, so it stays in the membrane. And the name of this molecule, it has 2 fatty acids. Those are in organic chemistry. They're called acyl, A-C-Y-L groups. Anybody had organic here? Okay, acyl groups. Okay, two acyl groups attached to a glycerol. So we call this diacylglycerol. Oops. Or DAG for short, diacylglycerol. It's extremely hydrophobic, so it stays in the membrane. Now, this molecule, this is anything but hydrophobic. It's about polar as you can get. You have inositol and 3 covalently attached phosphate ions. So we call this very polar molecule inositol triphosphate, often called IP3. There's a couple other abbreviations for that too. But what we've just done, folks, is we've taken this membrane phospholipid, which is a minor component of membranes. We've cut the polar head group off, forming inositol triphosphate. Meanwhile, the hydrophobic tail portion, the diacylglycerol, is left behind in the membrane. So let's do this over here. So we have HIF2, that's our, and we cut it into diacylglycerol and inositol triphosphate. The minute the inositol triphosphate is formed, of course, something that polar and electrically charged to boot, the last place in the world it's going to want to be is inside a cell membrane. So it gets out of the cell membrane like a bat out of you-know-where and goes right into the aqueous cytoplasm. Now the cool thing about this, folks, both of those molecules are cellular regulators. They activate things inside cells. So let's see what each of these guys do. Well, let's look at the diacylglycerol first. It stays in the membrane, and lo and behold, it binds to another membrane-bound enzyme and activates it, or family of enzymes. And that family of enzymes is yet another family of membrane-bound protein kinases. We recall the protein kinase A's and the protein kinase G's. Those were activated by cyclic nucleotides. This is the protein kinase C, or PKC family. And it is activated, among other things, it is activated by diacylglycerol. So now we've activated this kinase, and this thing goes around and phosphorylates targets. Most of its targets are other membrane-bound proteins, things like transport systems, active transport, facilitate diffusion, the like, other signaling pathways, PKC, certain PKCs can activate other signaling pathways. But it can also do things, proteins that would approach it from underneath, from the cytoplasm and bind to it. Questions there?
What's the possible?
Where? Oh, phosphorylates. In other words, that's what kinases do. They covalently attach phosphates to other proteins. So we get a lot of protein phosphorylation courtesy of activating protein kinases. Okay, meanwhile we have our very polar inositol triphosphate, or IP3. What does this guy do? It stays in the cytoplasm. But what it does is it binds to certain family of ligand-gated calcium facilitate diffusion channels and causes the channel gate to open. And those channels are located in things like the endoplasmic reticulum or ER membrane, to a lesser extent the mitochondrial membrane. And when it binds these things, and here's a membrane, here's a channel, IP3 binds to it and opens the channel. And what happens is organelles, now recall we said calcium levels in the cytoplasm are very low. They're about 0.1 to 1 micromolar. Normally a little bit closer to 0.1 micromolar. That's a 10,000th of what you're going to find outside. But and we keep calcium low because calcium ions activate all kinds of things in cells that you don't necessarily want to activate all at once. Now, if we want calcium into cells, we can open up calcium channels in the cell membrane. Calcium from the outside rushes in. But just in case, we do store millimolar quantities of calcium ions in certain vesicles and certain organelles. The endoplasmic reticulum is a good example of that. And we're talking millimolar calcium. So there's lots of calcium in here, very little in the cytoplasm. What happens when IP3 binds, it opens up the channel, calcium ions rush into the cytoplasm and do all kinds of things, either directly affecting proteins or activating calmodulin, and that goes and affects a whole bunch of proteins. So now we have a situation where we flood the cytoplasm with calcium ions, thanks to our IP3. And then the other thing is we activate the family of kinases, the protein kinase C families. So we have got numerous bases covered by this pathway. Yes?
So the IP3 just like makes it open. So
Yeah.
So it can
Yeah.
come out.
Yeah, the channel
It
is...
doesn't go in.
No, it doesn't. The channel is a ligand-gated facilitate diffusion channel. In other words, it's closed until a particular molecule, IP3, binds. So it binds on the outside, doesn't have to go on. Remember the neurotransmitter receptors that we talked about, neurotransmission? Same thing, the neurotransmitter never enters into the dendrite itself. It binds the channel. It binds the receptor, causes the channel open, and depending on the channel, ions rush in or rush out, depends on what the channel is. So this is the same kind of thing. In fact, it's kind of like, it acts like a neurotransmitter receptor, except it's not a neurotransmitter here. Okay, and it's facilitated diffusion, high to low concentration. Stuff just rushes out. It's an exergonic process. Okay, so we can do all kinds of cool things with this pathway, which is why it's widely used in a number of physiological and other types of events. And that kind of covers the G protein linked receptor pathway. Once again, many different receptors and many different ligands and many different trimeric G proteins activate this pathway. There's a whole bunch of different receptors, many different versions of G proteins. There are several kinds of phospholipase Cs. There's a whole family of DKCs floating around. So we have all kinds of, we can have all kinds of variations, all kinds of different processes occurring using the same basic signaling pathway. So it's widely used in all kinds of things. We've got everything covered here. All right. And once again, the appropriate, not all of them, but some of the PKCs, when they're activated, they can go on and activate other signaling pathways as well. Recall, even though we're looking at them separately, signaling pathways do interact with each other. You affect one, you may ramp up or ramp down others. Okay, any questions on that?